And so the final outstanding question that I'll spend some time talking about is what is actually happening to NAD turnover flux here at the bottom. So we know steady state is decreasing with age, but there are different ways that can happen. And just measuring the steady state doesn't really tell you whether NAD is being turned over faster or slower, or so are NAD-dependent enzymes actually more active or less active. So to try to get at this, we employed a system that was pioneered by a couple of different people, particularly Ling Liu and Josh Rabinowitz's lab, and then Melanie McReynolds when she was in Josh's lab, and Karthi Chalapa in my lab, used this system to measure NAD turnover in a variety of systems. So what this uses is this tetradeutyrated nicotinamide. So there's deuterium at 4 different positions. And when you convert this into NAD, this position at the top here is the redox active position. And so that starts exchanging and you lose that deuterium pretty quickly. And so you get mass plus 3 instead of mass plus 4. And that's shown over here looking at the NAD fraction in cells over time. Mass plus 0 is the original NAD the cells had. That goes away if you're incubating them with this labeled nicotinamide, and you accumulate mainly mass plus 3. You also can slowly lose a little bit of this deuterium as well. And that's accounting for this mass plus 2, which is a minority component that we don't usually pay too much attention to over shorter experiments. And so when we use this system, we can learn a couple of things pretty quickly in cell culture. In this case, we've just inhibited NAD synthesis and watched the decay of the unlabeled NAD. So the label here is really just showing you that we're not making any new NAD from the labeled nicotinamide that's present now. And what you can see from the decay is it follows approximately first order kinetics, meaning that the amount of NAD present is determining how fast it gets used up. So the enzymes that are consuming it are sensitive to the concentration, which means that if you boost NAD levels correspondingly, you would expect to increase the activity of some of these enzymes. On the right, we can show that in these cultured cells, which were a breast cancer-derived cell line called T47D, a lot of the consumer activity seems to be dependent on sirtuins and PARPs, which are some of the well-known consumers, because we can inhibit either one of those. And this F out is the turnover rate, essentially, of the NAD pool. And you can see it decreases pretty dramatically with either PARP or sirtuin inhibition, and about 60% of a decrease in that turnover rate if we inhibit both. I should mention these cells don't express CD38, which is the other major consumer people think about a lot. So we really don't know what's accounting for the rest of the turnover in these cells, or how relevant this conclusion really is to in vivo tissues. And I say that partly because if we look in vivo, one of the first lessons we got is that it makes a huge difference whether you're in vivo or not for some of the tissues. So cell lines derived from intestine here in gray, or cell lines derived from anything really, have a turnover half-life of around 6 to 8 hours for NAD. But if you look in vivo at the intestine, the half-life is about 20 minutes. So much, much faster. And we don't really still know why. The turnover in vivo is so much faster than in the cell lines, but you can see discrepancies in other tissues, and in particular muscle. The half-life speeds up in cultured cells, but it's actually about 14 hours in vivo, so very slow turnover of the muscle NAD pool. So of course we wanted to use this system to ask why NAD is lower in old mice. So we set up essentially the same thing that I've been showing you data from, where we have free-running mice that are getting constantly infused with this tetradeutyrated nicotinamide, now comparing 3-month-olds to 25-month-olds. And we really had 2 major models in mind, just to set up the way we're thinking about this. The first is that you might have decreased NAD because synthesis actually gets decreased with age, so you're not making as much. In that case, consumption would have to slow down to match the new lower synthesis rate, and this would lead to a decrease in NAD flux. Alternatively, you could have an increase in the activity of NAD consuming enzymes, which would lower the NAD pool because you're consuming more of it. And initially that would result in an increase in turnover. But as the NAD concentration falls, 2 things can happen. One, synthesis could be upregulated to compensate, and that would maintain the new higher NAD turnover flux. Or second, if synthesis does not cooperate and doesn't change, then what would happen is the new higher NAD consumption will lead to the NAD pool size shrinking to the point where now, because of those first order kinetics, the lower amount of NAD means there's less consumption by the consuming enzymes, and that now balances with synthesis again. And so in this case, flux overall isn't necessarily going to change very much. You might just see what looks like a change in the pool size with very little change in overall turnover flux. And so first of all, the first thing we looked at was nicotinamide mass plus 3. So this is nicotinamide that has been through a cycle of getting converted to NAD. So it lost one of the deuteriums and then was released again. So this is sort of an index of whole body NAD turnover. And we're looking in the blood at plus 3. And the first thing we saw was that in the aged mice, there's at least as much M plus 3 production. In fact, it looks like statistically significantly more, albeit not too much more, mass plus 3 production in the aged mice. So that just tells us right from the get-go that it seems like whole body turnover flux is preserved or maybe even accelerated a little bit with aging, not decreased as we would think if it was impaired synthesis. And I'll blow up one tissue here as an example to really kind of illustrate what we think we're seeing. So this is mass plus 0 NAD counts in skeletal muscle. And you can see in the old animals, there's less NAD at steady state, as we had seen before and as we expected. If we look only at the newly synthesized NAD, which is the labeled, the mass plus 3, you can see that's actually very similar. And maybe just a little bit more of it appearing in the old, which would suggest that the synthesis rate, if anything, is slightly faster in the old, even though the old actually has less NAD present. So again, arguing for increased turnover flux. Sorry about that. And if we look at it in terms of fractional labeling here on the right, we can see that the total fraction of the NAD that has been newly synthesized in the old, because it's a smaller pool, and there's a slightly more mass plus 3, the actual fraction that's newly synthesized is much higher. And so the overall conclusion from this is that the smaller pool in the aged animals is turning over more quickly to compensate. So really you're maintaining probably turnover flux. And we can show that on the next slide. So this is the turnover flux expressed in molarity in different tissues in the aged versus young animals. And so in many of the aged tissues, remember the pool size is smaller, but those pools are turning over a little bit faster to really maintain the flux, or in some cases be slightly higher, like in liver here or in spleen. It's a little bit higher in the aged animals. But overall, the main lesson here is that flux is maintained in the aged animals. It's not decreasing, which is what you would expect if it was synthesis that was impaired. It really seems to be that in the aged animals, the model that best fits the data is that consumer activity is increased, and that results in the pool size falling to a point where it balances out consumer activity with synthesis, and you can sort of restore homeostasis. So the last topic I'll talk a little bit about are why such high doses are needed in the rodent studies. Most studies are typically using about 500 milligrams per kilogram of NR, which is about 35 grams a day for a 70 kilogram human if you just extrapolate from body weight. Human studies are usually using about 1 gram, not 35, and that's justified based on arguments about allometric scaling, which is the argument that drugs are typically metabolized much more quickly in rodents, and so you can get away with lower doses as the equivalent for humans. And the fact that there is toxicity that's seen with nicotinamide above about 3 grams a day in humans. And so there's definitely a fear of going to these much higher doses. But human and mouse cells have about the same NAD concentration and the same turnover rate. And so it may well be that you actually have to scale by body weight if you want to get the same effects that we're seeing in the rodents. And so to really try to get a better idea what's going on in vivo, we've employed this tool synthesized by Marie Magot, who's a chemist at the University of South Alabama. And she generated nicotinamide riboside for us that had a label of deuterium on the ribose ring and a C13 on the nicotinamide moiety. So we could track both rings separately. And if this goes straight into NAD, we'll get mass plus 2. If it breaks down, we'll get mass plus 1. And so when we do this, we have delivered NR or NMN either intravenously or orally, and then done a time course looking at first nicotinamide in the blood. So you can see a lot of it breaks down to nicotinamide, but you get a lot more if it was given intravenously than if it was given orally with either molecule. And then if we look in vivo at tissues, you can see here first, if you look at kidney, this green color is the MAS+2. So this is the molecule that we administered going directly into the NAD pool. And you can see a substantial amount in the kidney with either NR or NMN given intravenously. Not very much, just wisps, if anything, of MAS+2 when we've given them orally. If we give NR IV, we get the same thing in muscle where you get lots of MAS+2 contributing. With nicotinamide mononucleotide IV, we actually don't see that. It seems like it contributes much less to muscle when it's given intravenously. And if we give them orally, we really don't see any MAS+2 appearing at all. And so there's a couple of lessons here. One of them is the delivery route makes a huge difference. So I'm losing control of my slides here. But the delivery route makes a huge difference. Intravenous is clearly much more effective at getting these molecules intact to certain tissues. The second lesson is that there really seems to be something missing when we look at the oral pools. We're not really accounting for some of the administered molecule. It seems like there's less of it everywhere. And one of the things we've recently discovered that might be contributing to that is that the microbiome is playing a huge role in converting nicotinamide that is administered orally to nicotinic acid, or the same thing when you give nicotinamide riboside. You see, so this is the spike in blood levels of nicotinamide here on the left after giving oral NR, and you can see we get a pretty good rise into the 100 micromolar range. And on the right is nicotinic acid, and this is not produced by any mammalian enzyme, but is completely microbiome dependent. And you can see this is actually getting up into the tens of micromolar and 70 micromolar range as well. And that's a high enough concentration to activate G protein coupled receptors that bind nicotinic acid, and to potentially have effects on other enzymes that are independent of NAD synthesis. And certainly it's enough to dramatically fuel the Priest-Handler pathway to make NAD directly from nicotinic acid, which is a different set of enzymes and may have different effects than we've realized from only looking at the NR or NAM on cells. So we think this is really important to follow up on and understand whether nicotinic acid production in humans receiving nicotinamide riboside is in this range. So to really get at this in a little more detail, Marie was kind enough to synthesize an even more advanced derivative of nicotinamide riboside for us. So this is a +9 that has all the ribose labeled, so we can track that ring, but then on the amide group labels the oxygen, nitrogen, and carbon. And so we're able to see if this has been deamidated to nicotinic acid by the microbiome and then resynthesized into NAD. We can tell by the mass whether that's happened, even if it's already back in nicotinamide form when we're looking at it. And so Carthy orally dosed mice with this compound, and there's a couple of lessons from this experiment. The first one is if you just ignore the labeling patterns at first and look here at control mice dosed with nicotinamide riboside. This is liver NAD levels, and you can see they go up about 3-fold with nicotinamide riboside. If the mice got antibiotics first, so they don't have a microbiome, then nicotinamide riboside doesn't really work so well. We get a little bit here of MAS+9 in purple. So there's a little bit of a rise, but it's not even statistically significant anymore if they've had antibiotics on board. And instead, the majority of this NAD boosting effect of nicotinamide riboside is coming through these MAS+3 and MAS+1 forms, which are the molecules that show that the nicotinamide riboside broke down, was deamidated by the microbiome, then was taken back up and converted to NAD. And so there's this huge effect that was not really recognized that is being played by the microbiome when you take these supplements orally. And a big part of them is going through a nicotinic acid intermediate rather than staying as the parent molecule or even nicotinamide. So this has some important consequences because a big part of the argument for using these things has been the energetic efficiency. So this is the energetic cost of going through the standard salvage pathway from nicotinamide, cost about 4 ATP. If you give nicotinamide riboside, you can get there with about 2 ATP because of this nicotinamide riboside kinase, which is a more efficient reaction. And when we give molecules intravenously, this is kind of what we see. The majority of it does go through the nicotinamide riboside pathway as anticipated. We see some going through the NAM pathway. But when we give things orally, which is the main way they're given to humans, you can see that there is actually, well, first of all, a lot of it's unaccounted for. There's less going through nicotinamide and less going through the nicotinamide riboside. In fact, very little going through directly through NRKs, through this green pathway. And instead, we're actually realizing that the majority of the delivered bolus is going through nicotinic acid through this alternative Priest-Handler pathway to get to NAD, which is actually energetically less efficient. It takes more ATP to go this way, and may have other consequences, as I alluded to, in terms of activating G protein coupled receptors and things like that. So I think this is also really going to be important to the interpretation of human studies using NAD precursors to really understand how this metabolism is all working. In addition, this got us looking at tracers in mice related to the microbiome. We ended up doing some studies that suggested that even when you don't give this orally, when you give it intravenously, you can end up with label being converted by the microbiome. And so we did this experiment where we didn't actually administer any exogenous precursor. We're just looking at normal metabolism now. And we've generated mice that were antibiotic treated, so they have no microbiome. That's the ABX. And they're on a diet with or without nicotinic acid present. And we're measuring nicotinamide in the colon lumen. And you can see here, when you wipe out the microbiome with antibiotics and take all the nicotinic acid and nicotinamide out of the diet, you actually end up having an increase in nicotinamide that's present in the colon lumen. And we see the same thing with germ-free mice. So these are mice that were ex-germ-free, that were given bacteria again on the left, and then compared to germ-free mice looking at the colon lumen, there's higher levels of nicotinamide in there. And so it seems like the microbiome is actually consuming nicotinamide, and we really at this point had no idea where it was coming from. So we did a series of experiments to show that it actually is coming from the bloodstream, from the host. It's not present in the diet. There's no microbes there to make it. That was really the only explanation that was left. And so we did intravenous administration of this deuterated nicotinamide form, and then looked in the colon lumens, the ileum and so the small and large intestinal lumens of mice that were, again, germ-free is the GF here. So these are germ-free mice that were not administered on the left, that were administered the tracer on the right. And you can see that tons of nicotinamide gets into the colon lumen from the bloodstream. If we've conventionalized the mice, this is decreased quite a bit, how much nicotinamide we see there, because it's being metabolized by the microbiome. Kind of the same thing in the large intestine. And the reason that we're seeing this nicotinamide disappear in the mice that are conventionalized that have a microbiome is because the microbiome is converting it all to nicotinic acid. And so here you can see, again, nicotinic acids piling up in those mice after intravenous administration of nicotinamide. And that nicotinic acid contains the label. So we know that it's actually coming from the intravenous nicotinamide that we administered into the bloodstream, and it's getting secreted into the colon lumen, then converted to nicotinic acid. And so this is the cycle that I essentially just described. So conventionally, we think that nicotinamide or nicotinic acid come from the diet, and the microbes might get a chance to interconvert them while they're in your gut lumen, and then you would just absorb both and have them in the body as vitamin B3. We do know that you can absorb nicotinamide, but here we show that really the net flux under normal conditions is out. There's more nicotinamide going into the gut lumen than being absorbed from the gut lumen. And in fact, it's generating this cycle where nicotinamide gets secreted into the gut lumen, then converted to nicotinic acid, and then reabsorbed by the host. So there's a sharing of NAD equivalents between the host and microbiome. So finally, we were curious about how important this pathway is to microbial NAD synthesis. It wasn't really clear where microbes get their NAD precursors in the case where you take them out of the diet. And so we decided to test that by doing the same type of experiment. Now, instead of a bolus, we do constant infusion of intravenous deuterated nicotinamide, so we can track this into the gut lumen. And so here you can see steady state levels being reached for NAD labeling in the microbes, which are the main NAD content of the gut lumen. In the small intestine, it's a little bit higher than in the colon or in the stomach, but in all cases, pretty significant. And if we take into account the serum labeling of nicotinamide shown here on the left, which is about 40% on average or so, reaching about 50% by the end of the experiment. If we account for that, we can calculate that about 80% of the NAD present in microbes in the lumen of the small intestine actually originated as nicotinamide in the host circulation. And so this really seems to be the major pathway that's providing NAD precursors for the gut microbiome. at least in the small intestine, and a major pathway in the large intestine as well, where we see a lot of the balance being made up by synthesis from fiber. And so just to go through some conclusions, I think that boosting NAD is an effective strategy to treat many conditions in mice, but studies in humans to date have shown weaker or no effects for some of the same indications, although positive effects in things like mitochondrial myopathy. And so we think it's a very exciting time to be looking at human studies, but also to be thinking about why we're not translating all of the things that we see in mice into humans yet, and a better understanding of some of these pathways that are relevant to NAD metabolism might really help us get there, considering things like intravenous dosing, or whether we really need to be using higher doses or local doses for certain indications, and really what the downstream pathways are that are relevant, which will be really important if we're really going to achieve some of the goals we have in humans for these NAD precursors. So clearly there's a lot left to learn about the fundamentals of NAD metabolism. And so with that, I'm done. I was asked to comment a little bit on my own habits with regard to supplements and NAD precursors. And I can say I don't actually have a regimen that I have adopted or committed to. I certainly experiment with NAD precursors once in a while just to see if I can feel any of the things I've heard anecdotally from people. I can tell you, for the most part, they've only made me not sleep as well. I feel like I have too much energy at bedtime sometimes if I've been taking them. But again, I don't really do anything consistently at this point, other than I also do tend to try to eat in a 12-hour window, which is more just my habit. I feel better when I do that. I really think, to be honest, that we have a lot more research to do with respect to NAD precursors to really be able to firmly commit to a science-backed regimen. And I hope that we're going to be able to get there. So with that, I just want to thank some of the people in my lab, and particularly point out Karthi Chalapa, who did a lot of the tracer studies that I showed you in collaboration with Melanie McReynolds. Tim Luongo did a lot of the mouse work and the mitochondrial transporter studies. I showed some data from Shamistha Mukherjee on liver regeneration. And of course, everyone here made major contributions to the work coming out of the lab over the last few years. And so thank you for listening. I hope you enjoyed the talk.